Good morning. This is a presentation on the history of alkaptonuria, a timeline of the past to the present and to a possible future. We begin in ancient Egypt. This is the mummy of Hava, who died in his early 30s. He was a custodian of a granary in Egypt in 1500 BC. The discoloration you see on the mummy is not alkaptonuria pigment, it's the mummification process. However, when the mummy was subjected to X-ray and other investigations, they found extensive calcification in the spine, as well as joint disease in both the hips and knees. As a result, alkaptonuria was suspected, and a needle biopsy of the hip was carried out, which showed that Hava had homogenic acid pigment in his hip joint. This confirms the diagnosis of alkaptonuria as having been present even in ancient times. And the question is, how common was AKU in ancient Egypt? I suspect there might have been quite a lot of consanguinity in those days. When you look at the consanguinity rates of the Middle East countries at present, you can see Egypt is one of the highest at 21 to 80%. It is therefore not unlikely that consanguinity was quite prevalent in ancient Egypt as well. When you look at the prevalence of AKU in the UK, it's about 1 in 250,000 to 1 in a million. However, even within the UK, the prevalence varies enormously, and a newborn screening carried out in Wales between 1970 and 1977, where nearly 250,000 infants were screened, they found 6 cases of alkaptonuria. So the prevalence, the incidence is more like 1 in 44,800, which is 6 to 20 times higher than in the rest of the UK, and reasons for this are not clear. We now go to the Middle Ages, and around 1580s, there were at least 3 cases described of black urine. So Scribonius in 1584, who described a healthy schoolboy who continuously excreted black urine. Similarly, Schenk in 1609 described a monk with similar black urine. And Lusitanus in 1649 also described a 14-year-old boy who passed black urine. And the Lusitanus one is particularly striking because he describes it as, at the age of 14 years, he was subjected to bleedings, purgation, baths, a cold and watery diet, and drugs galore. So you can see they were interested in curing AKU even then. None of these had any obvious effect. The patient therefore got fed up and resolved to let things take their own natural course. None of the predicted evils that were suggested to him ensured. He married, he had a large family, and lived a long and healthy life, always passing urine black as ink. Although it says he lived a long and healthy life, I wonder what the lifespan was in those days. It may have been fairly short. We now move to the 19th century, when a lot of developments in AKU occurred. We start with Bodecker, a German scientist who was the first to describe a modern AKU patient as having an unknown substance. He realized when he was testing the urine that the substance was a reducing agent. It reduced failing solution, namely blue copper sulfate to red copper oxide, but not bismuth hydroxide, unlike glucose. So it was called the Nylander's reaction. So in other words, glucose reduces failing solution and Nylander's reagent, but the black urine only reduced failings but not Nylander, so not diabetes. This is the failing solution. The color change, as you can see, from blue to red. And this is the Nylander's reaction where there's a black precipitate of bismuth black. Bodecker also made another important observation. He realized that there were urines which were black when passed, and that there were urines which became black on exposure to air. And you can see here, I've reproduced what he did in the laboratory here by adding sodium hydroxide to the same urine in the two test tubes. Only the tube on the left has sodium hydroxide added along the side of the test tube carefully so that it doesn't mix with the urine as a whole. And when you do that, you can see that the urine darkens from the surface. And Bodecker realized that this meant it took up oxygen, which is present at higher concentration nearer the surface than in the body of the urine, and under alkaline conditions too. So for this reason, he called this alkapton, after its behavior towards oxygen in alkaline solution. Before he could chemically identify the alkapton, the patient discharged himself. In 1866, Virchow, an eminent histopathologist, reported the first case of pathological curiosity of an AKU patient. He carried out an autopsy of a 67-year-old man who died with generalized edema, or anasarca. We don't know whether the patient had aortic stenosis and heart failure. The subject also had deforming arthritis. Internally, the cartilages and the tendon insertions all over the body were stained black to light gray. Under the microscope, and even visually, the tissues were yellow in color, or ochre, and for this reason, Virchow called this ochronosis. Of course, he did not know at the time that alkaptonuria and ochronosis were the same condition. We now move to 1891, and some important German scientists, Volkow and Baumann, they identified that alkapton was 2,5-dihydroxyphenylactic acid and named it homogentisic acid, the next higher homologue from gentisic acid. Of course, gentisic acid is metabolite of salicylic acid, and salicylic acid, of course, is the precursor of aspirin. He showed that homogentisic acid also came from tyrosine, and he and his colleagues debated whether this was the accumulation of a metabolite before a block step of normal metabolism, or whether it was an abnormal compound formed by an abnormal series of reactions. They also went on to describe the series of reactions leading to homogentisic acid. They got so much right, but what they got wrong was that they stated categorically that homogentisic acid was formed by putrefaction of protein in the gut and not by tissue metabolism, because they were convinced that only plants and not animals could make aromatic compounds from non-benzene structures. We now move to 1899, when Hecker and Wolf published eye ochronosis findings for the very first time. And this is the first time that external ochronosis was linked to alkaptonuria. We now move to 1902 to Archibald Garrod. He had 28 cases of alkaptonuria, and he was working at the time at Great Ormond Street Hospital in London as well as St. Bart's. It is fair to say that he unraveled alkaptonuria, the prototype inborn error of metabolism. He was also working on other rare diseases like albinism, pentacyria, and cystinuria. But it was working with alkapnuria that he uncovered the mode of transmission of this first hereditary human disease. He also elucidated the first intermediary metabolic pathway and introduced a link between a specific enzyme and a specific Mendelian factor, which are now called genes. So he is rightly considered the father of inborn error of metabolism. Albrecht is the first person who put together the connection between alkapnia and ochronosis as the same disease. He autopsied a patient and also reviewed 6 other autopsies that were carried out previously and made the connection. He also noted that all of these cases also had deforming arthritis. Interestingly, Garrod was not interested in ochronosis or the consequences of ochronosis. William Osler first described the ochronosis in the ears and skin externally for the first time. So by now we had the eye ochronosis, ear ochronosis, and the skin ochronosis recognized in alkapnuria. We move forward 30 years, and it's Neubauer who described a monograph 300 plus pages long, where he reviewed and deduced the pathway to homogenic acid, including all the enzymes. Further 30 years later, Ladou and colleagues in the National Institute of Health studied a liver biopsy from an alkapnuria patient and showed that all the enzymes of the tyrosine metabolism were present in the sample, but that homogeneous oxidase was inactive. And this was the paper that was published in the Journal of Biological Sciences, Journal of Biological Chemistry in 1957. And you can see the National Institute of Health has always had an interest in alkapnuria, and come back to that later. Move forward another 30 years, and a development was occurring, not directly related to alkapnuria at the time. Edward Locke and colleagues developed nitisinone from leptospermone from the Australian bottlebrush plant. So the nitisinone derived from leptospermone was to be used as a herbicide, and this was developed by Imperial Chemicals Industries, ICI. Before it could be used as herbicide, they did the mandated toxicology tests, which showed that many animal species developed keratopathy on nitisinone. Investigation of this unwanted effect showed this to be due to high tyrosine, which in turn led to the deduction that nitisinone inhibited the enzyme hydroxyphenyl pyruvate oxidase in the tyrosine pathway. Professor Lindstedt and Professor Elizabeth Holmer were working with hereditary tyrosinemia type 1, and they realized that this inhibition of HPPD by nitisinone could be of value as therapy in this life-threatening disease. Children didn't live much beyond 2 years of age due to liver failure and liver cancer. Until then, the treatment for HD1 was liver transplantation. When they tried nitisinone with Edward Locke's help, the child that was given nitisinone improved dramatically. Within 48 hours, the liver profile test improved, the abdominal protuberance decreased, the hepatomegaly resolved, and the child got well. So since then, the standard of care for HD1 is nitisinone. In 1993, Pollack and colleagues in Boston, USA, through heterozygosity mapping, showed that the AKU gene was present on the long arm of chromosome 3. Something important happened in 1998. A Japanese group carried out a novel therapeutic trial of nitisinone in alkaptonuria murine model, and they showed that the nitisinone could decrease homogenetic aciduria in this murine model. So they must have been working on it for a time before the publication in 1999. In 1998, a group in the NIH also proposed that rather than waiting to cure AKU with gene therapy, a potential treatment was already possible through the use of nitisinone in alkaptonuria. And of course, between 1998 and 2008, the National Institute of Health carried out a series of elegant studies culminating in a 3-year randomized clinical trial of nitisinone in patients with alkaptonuria. And I'll come back to that in a minute. So in 2002, nitisinone, which was acquired by Swedish Orphan, was marketed as Orphanin and approved for hereditary thalassemia type 1 in the US in 2002 by the FDA and by the European Medicines Agency in Europe in 2005. In 2003, we are now moving back to AKU as the focus. A patient founded the Society for Alkaptonuria in 2003, along with the doctor, which is myself, as well as with Nicholas Sineru as the CEO. This created an opportunity for AKU patients for the first time. At the time, there was no real opportunity for people with alkaptonuria in the UK or anywhere else. The first piece of research that was carried out in this collaborative work between the AKU Society and the Royal Liverpool University Hospital was a post-mortem study of a patient who donated a body for AKU research. And this study raised novel research questions which gave us our direction. In 2005, the United Kingdom AKU population identification study began. We did this because we wanted to be ready to be able to administer nitisinone to our UK patients when it became available following the NIH clinical trial. So we began a population identification program between 2005 and 2009. 16,000 GP questionnaires were posted with a 16% response. More importantly, our patient numbers increased from 4 at the start to 79 at the end of the study. At the same time, we were also understanding more about the pathophysiology of alkaptonuria. Professor Gallagher at the University of Liverpool led on these models to increase the understanding of alkaptonuria, and of course the cell model for achronosis was developed in 2008. There was bad news in 2008, which is that the NIH 3-year nitisinone trial in alkaptonuria reported inconclusive, and therefore the FDA did not approve nitisinone for alkaptonuria in the US. In order to understand the reasons why the trial failed, because we thought it might have been because the numbers were too small, they had only used 20 patients on nitisinone versus 20 patients not on nitisinone. We thought the dose response wasn't well characterized. We thought the outcome measure wasn't right because they only used lateral rotation of the hip to decide on the success or failure of the trial. For all those reasons, we undertook a study called the UK Natural History Study to better understand AKU in humans, as well as to develop better outcome measures for alkaptonuria for clinical trial. At the same time, in 2011, the basic science was progressing really well because by now we had a mouse model of achronosis, because until then it was thought that AKU mice did not develop achronosis. we proved that it could, and the fact that nitisinone could inhibit the development of oculosis in these AKU mice. So really important to confirm that nitisinone could be effective not just in lowering homogenic acid, but could modify the disease process of oculosis. 2012 was a landmark year for AKU because the highly specialized services took note of all the things we were doing in terms of improving AKU in the UK and commissioned a national service for alkapnuria at the Royal Liverpool University Hospital. So the National Alkapnus Centre became operational in June 2012, having been designated as such in April 2012. In 2012, we were also successful in being awarded €6 million with 4 million co-financing to develop nitisinone as treatment for alkapnuria for regulatory purposes. And this was called Develop a Cure. So by now we had two strands. One was the off-label use of nitisinone in the National Alkapnus Centre, which would give us real-world data. And of course, we now have the opportunity to do clinical trials in order to secure regulatory approval. And you can see that was approved in April 2012. This is the research consortium that undertook Develop a Cure, and you can see the sponsors were the Royal Liverpool University Hospital and the University of Liverpool. You can see the three study sites, the Royal Liverpool University Hospital, Hospital Necker in France, and the National Institute of Rheumatology in Pišťany in Slovakia. There were 4 laboratories. The Royal Liverpool University Hospital carried out all the metabolic studies. The University of Sierra, the inflammatory and oxidative markers, Nordic Biosciences, the cartilage and bone and tissue biomarkers, and the Bratislava Center, which carried out genetic analysis. Swedish Orphan Biovitrum was the pharma company, and PSR was the CEO expertise in the study. And not forgetting the 2 patient charities, the AQ Society in the UK and the AQ Society in France called ALCAP, who were responsible for the important and difficult job of recruiting and retaining patients. In 2014, the dose response study called SONIA 1, suitability of nitisinone alkapnuria 1, reported that the 8 milligram dose was better than 1, 2, or 4 in terms of reducing homogenic acid with no safety issues. In 2014, the 4-year clinical trial using 10 milligrams of nitisinone, which was a hard capsule, unlike the liquid nitisinone used in SONIA 1, began. 138 patients were recruited, 69 on nitisinone and 69 not on nitisinone. It's the largest clinical trial in the inherited metabolic field ever. We also carried out a cross-sectional study on oculosis called the subclinical oculosis features in alkapnuria, or SOFIA. And this showed in 2017 that oculosis could be present earlier than age 20. Therefore, early treatment of AKU is therefore needed since oculosis is not fully reversible. In 2019, SONIA 2 was completed, and this showed that nitisinone 10 milligrams lowered homogenic acid by more than 99%. Importantly, it also decreased oculosis. It didn't just arrest oculosis, but it also partially reversed it, which is important for people with advanced disease. And nitisinone also, more importantly, showed that there was significant slowing of the disease, and patients were better with less pain. and feeling more comfortable. This was the publication in the Lancet Diabetes Technology in 2020. By this time, we realized that although nitisinone was very effective in lowering homogenic acid, it also caused high tyrosine. So that required lifelong dietary management. So we were at the time interested in developing better treatment opportunities for alkaptonuria. But to understand where and how the HED replacement was going to have to happen, we needed to understand liver and kidney HED, the homogenic cytokinase enzyme, since these are the only two tissues that it's expressed in any significant quantities. And the upshot of the research led by Professor George Bugaris showed that HED in the liver is more important than the kidney, and that liver knockout of HED produces all of the features of alkaptonuria. In 2020, based on the SONIA 2 study, the European Medicines Agency approved nitisinone as the first treatment for adult patients with alkaptonuria. And this is the publication in the EMA portal. As I said, by now we were beginning to think about the unwanted consequences of tyrosinemia. For example, we knew that the tyrosine could precipitate in the cornea and cause a red eye syndrome requiring lifelong dietary management. We've also since realized that 5% of patients, especially those with autoimmune disease, can develop vitiligo while they're on nitisinone. Cataracts are more common both in untreated AQ patients and does not stabilize following nitisinone. So therefore, it's both homogenic acid and tyrosine causing the cataract. We also found that Parkinson's disease is 20 times more common in AKU, both before and during treatment. We think the high tyrosine can unmask Parkinson's disease. Low protein diet, which is needed for the tyrosinemia management, is inconvenient and has social consequences. Importantly, low protein diet also causes lower muscle mass, which is not great in a condition that requires surgery. So the future of the AK research is concentrating on developing co-therapies and new therapy development. And these are some of the leaders that you see in the photograph shown there. So the co-therapy with nitisinone will involve the development of tyrosine absorption inhibitors, and we are working with Sanofi and other pharma companies on that. We're also developing HED mRNA therapy, working with biotech companies and with philanthropic funding. We've just started a PhD studentship to develop HED gene therapy, and we're working with Sanofi and UCL on this. So this is a history loop of all the things that I've said so far.